Good morning, everybody, and welcome to the Department of Entomology seminar series. This morning, it is my pleasure to introduce Dr. Lyric Bartholomay, who is a professor in the Department of Pathobiological Sciences at the University of Wisconsin-Madison. Lyric is the director of the Midwest Center of Excellence in Vector-Borne Diseases, which is one of 5 CDC-funded centers in the United States. I'll just share a little bit of background here. So Lyric obtained her bachelor's from Colorado State and her PhD from University of Wisconsin before taking up appointments with the faculty at Iowa State, where she held positions as assistant and associate professor and remains adjunct to this day. In 2014, she joined the faculty at the University of Wisconsin-Madison, and she was kind enough to share her bio sketch with me, and her resume really reflects a very strong emphasis on professional service. She has held positions as chair of the American Committee for Medical Entomology, which is a part of the American Society for Tropical Medicine and Hygiene. She is currently a section editor for several journals, including the Journal of Medical Entomology and PLOS Neglected Tropical Diseases. And she's also the recipient of numerous teaching awards and awards in recognition of research and innovation. Her program combines both field and experimental practice in public health entomology with an emphasis on vector physiology, studies of vector pathogen and parasite interactions, including viruses and filarial worms, with work in vector-borne disease ecology, as well as applied technologies with an emphasis on mosquito control. So very broad experience. And today, she is giving a presentation on the Midwest Center of Excellence for Vector-Borne Disease, enhancing public health entomology capacity and practice in the Upper Midwest. So welcome to Lyric, and I will cede you the floor.
Excellent. Thank you so much for the invitation to be here to talk about our Center of Excellence. And as I told Kate earlier, my intent is to kind of give you an overview of what we've done with our center. And so I can't go terribly deep into any of the data, but I'm very happy to have conversations later with you if you want to talk more about some of our projects and about the projects that Kate was telling you about beyond the work I do in the center. Being a faculty member is the best job in the world, right? Because we can do all of these things and have this really diverse portfolio of research. So the Center of Excellence is one of 5 centers established in 2016 in response to the Zika outbreak that hit the United States, primarily in Florida, right? And so the federal government allotted a pot of money to the CDC to come up with a response strategy because what we learned in that outbreak was that we really had a catastrophic PR issue and capacity issue because we just didn't have trained medical entomologists to respond in Florida to the outbreak. So we even Kate probably got emails from the CDC begging us to go to Florida for 6 months to work on Zika because they just didn't have hands to do the work. And so they came up with these concepts for the Centers of Excellence to do a variety of things. First was to really build capacity. So increase the number of entomologists and medical entomologists in particular who were available and to deploy in these kinds of situations. They also wanted to better connect academia and public health so that there was this more formalized synergy between the research that we can do and public health need. And then they also really wanted applied research to better be able to control vectors. And so we put in a proposal from the Midwest, as did many people. And I think part of the reason that we were successful in getting our Center of Excellence was that we really have some serious issues beyond Zika, right? So obviously Zika is not an issue in Wisconsin, but because the monies were available and the capacity building was so important, the CDC had the foresight to say, let's not just tackle Zika with this, but let's also try and get some other vector-borne disease capacity built. And really in the upper Midwest, our struggle is primarily with tick-borne disease, but also with some arboviruses that are transmitted by mosquitoes. So our center includes a 5-state region, and it really extends beyond this. But the 5-state region where we actually have sort of subcontracts to our center award includes Minnesota, Iowa, Illinois, and Michigan. And so we have land-grant universities across this 5-state region who were already doing great work in public health entomology. We couldn't get the University of Minnesota, so we instead are working with the Minnesota Department of Health in Minnesota. And as the center has gone on, I mentioned we started in 2016, but as we've sort of progressed, states around us have joined our efforts too. And so we are contacted by and working with people in Indiana and Ohio and Missouri, sometimes in the Dakotas. But our award itself is really distributed amongst these 5 states. So we include these academic institutes and public health agencies, and also we work really quite a bit with mosquito control districts. So trying to understand what their needs are as well in terms of vector control. Excuse me. So our edict from the CDC when we got the request for proposals was to come up with an approach that I talked about, where we combine efforts in training, communities of practice with public health, and applied research, and all of those things synergize and interdigitate. And so that's what I'm going to sort of show you, how we've manifest these 3 goals for the centers. The disease burden, as I mentioned, in the upper Midwest is principally associated with Lyme disease, really. So the arthropod-borne disease of most concern is Lyme borreliosis. The numbers of cases of those just climbs yearly. The tick has expanded dramatically. And in Wisconsin and in Minnesota, we've seen really extraordinary work done to show that the black-legged tick, Ixodes scapularis, transmits a lot more than just Borrelia burgdorferi. In fact, there are 8 or 9 different pathogens circulating in our tick populations. Those include ehrlichiosis, anaplasmosis, etc. We also continue to have issues with arboviral diseases, so those that are transmitted by mosquitoes in particular, to include West Nile, La Crosse encephalitis, Eastern equine encephalitis, which I know has been a concern for Indiana, and a virus called Jamestown Canyon. So I'll talk more about these. But again, these endemic diseases were so pressing for public health that I think this helped us make the argument to the CDC that a center should be based at UW-Madison. Right, so I'm going to talk about some of the projects we're doing with ticks, and then about mosquitoes, and then about the work that we do with training. And in terms of ticks, we have a variety of different projects ongoing across our 5-state region, but surveillance was really important because we need to understand where the ticks are in order to understand disease risk. So in terms of surveillance then, we understood that ticks were in particular states and counties, and forgive me for having this color schema. I realize it's a little raw following up on the election to have all these blue and red counties. For some reason, this is the precedent in our field, is to mark counties as being either red as established, or blue as reported, or white as having no record. I'm thinking we should go grayscale. Anyway, with the data that were available back in 1996, we could see that black-legged ticks were sort of maybe in half of the state of Wisconsin. We weren't seeing them yet in Michigan, and just sort of sporadically in the surrounding states. As you see in 2015, with work that was done by Minnesota Department of Health and Susan Paskowitz's lab here at UW-Madison. I should say I should have said from the start that I'm a director of the Center of Excellence along with Susan Paskowitz. So the 2 of us are the directors, and by working together, we can really cover mosquito and tick biology. So Susan's lab here in Wisconsin showed that the tick was more widely distributed across the state. And then in Michigan, Jean Sau's lab was showing that the tick was invading Michigan. And because of the center and our capacity, then we've done a lot more comprehensive work, and that we've shown that up to 79% of the counties in our 5-state region are now supporting established populations of Ixodes scapularis. This is really important because physicians need to know that ticks are present and risk exists. So we hear from people in counties across Wisconsin where they've talked to a physician and the physician says, well, we don't have the tick here. But in fact, the tick is here and it's here to stay. So these help with risk assessment. We're also really interested in not just where the ticks are, but what it is that makes for a tick-human interaction. So the ticks might be present in a given county, but that doesn't necessarily mean that the tick is going to attack a person, right? There has to be behavioral things happening with that person in order for them to come into contact with a tick out in the environment. You know, these aren't flying vectors, right? They're just lying in wait with their front legs up for somebody to walk by and get swept up and onto a host. And so what human behaviors sort of precipitate those interactions with a tick? We've come up with a variety of ways to try and tackle this question. And one of them is that we've recently done a 3-state study on tick-borne disease prevention. We sent out tens of thousands of surveys across a 3-state region in counties where we knew that there were established black-legged ticks. So we did this in Wisconsin, Minnesota, and Michigan in concert with Jenna Bjork, who's now one of the directors of the Vector-Borne Disease Unit at the Minnesota Department of Health. So after a year of deliberating about what questions in particular to ask, we came up with a set of questions that dealt with things like, what do you know about ticks? What do you know about disease risk associated with ticks? How much would you be willing to pay if there were ticks in your yard? Would you be willing to pay a little extra property tax if the city would control ticks for you? Those kinds of questions. And so after several months of sending out postcards and soliciting responses to our survey, we ended up getting 2,836 surveys completed across our 3-state region. Wisconsin, you can see we hit our target of 1,000 responses. That's because we sent out rounds of postcards to really encourage people to participate. And we gave them Walmart gift cards, which was an incredible fiasco. So I'm very glad that this survey is done, because this was a whole beast of an activity. Overall, we got the large majority of people that responded to our survey. Sorry, my mouse is a little finicky. Were white and tipping toward a little bit more female than male. Many of them had a 4-year degree. 20% of them were earning $100,000 annually, and 37% lived in a rural setting. So you can see we have a biased sample, even though we did try and provide the survey in such a way that we would cast a broad net. We asked where people were, where they were and whether they would be willing to perform tick checks, if they used repellents, if they would treat their property with a pesticide, or if they treat rodents on their property with pesticides. And actually saw in the upper Midwest that a lot of people were performing tick checks, or at least they were saying they did tick checks. And about half of them were wearing repellent, which is probably not surprising. But the interesting thing is a similar study was done in Connecticut and Maryland a few years back by Allison Hinckley and others, and they found what looked like maybe some differences in human behaviors between the upper Midwest and the Northeast. And there's a difference in the invasion status of the tick, right? So it could be that part of those differences are associated with the ticks being newer in the upper Midwest as compared to on the East Coast. So another activity that we took on when we started our center was we wanted to also better understand individually what was happening with people and what they were interested in when it came to ticks. We created something called the Tick app, which you can download on Android or iPhone platforms. It was a joint effort between our center and the Northeast Center for Vector-Borne Disease at Cornell, and a really remarkable group of people in the Center for Health Enhancement Studies here at the University of Wisconsin. We created this app to collect data to understand when are people really coming in contact with the tick. So the kind of prevailing sentiment is, if you go for a hike in the spring in the upper Midwest, you may come into contact with a tick. But what we're finding is that the tick exposures may go well beyond just those people that are going hiking on a spring Sunday. So we ask people to download the app. We recruit broadly as possible, send out flyers and contact colleagues and different agencies where people we know may be working outdoors. And the Tick app has evolved. So we put it out for the first time in 2017. And we had a variety of features where we asked people to make a diary and report a tick. We gave them information on tick safety and how to identify a tick. Then in 2018, we wanted to get more people responding, so we sort of gamified and incentivized people's participation by giving them badges and little pop-ups that were congratulatory. These are things that evidently in the gaming world are useful, a whole science that I know very little about, but it's been really interesting to be exposed to. And so with the evolution of the Tick app into 2018, we were doing what was called a daily log instead of a tick diary. So we kind of changed the lingo. We asked people to report ticks if they came in contact with them. Essentially what we were doing was we were trying to get people every day to respond to a series of questions to ask where they had been and what kinds of things they had been doing, what kinds of activities. And so then we would understand if they reported a tick, what activities they were doing when they came into contact with that tick. Because I think we do need to get to this finer scale of timing when people are actually coming in contact with questing nymphs. In 2019, maybe I have this off. This could be 2018, 2019, 2020. But anyway, as the Tick app has evolved, we then added some new features. Every year we were asking our users what works, what doesn't work, what's fun, what would make you participate more. And also then we wanted to add in our latest iteration, so this is actually 2020, was preventive messaging. And we were getting people enrolled to use the Tick app. This does require IRB approval. And we're getting people in to use the Tick app. And then we were also dividing them up in such a way that they would get alerts to different tick prevention behaviors. So some of the people who were using the app would get a pop-up message that would say, if you've been outside, take a shower. So one of the things that we're thinking as a community is that perhaps showering after being outdoors would give you opportunity to sort of scrub the ticks off of you before they can get attached. So try showering, or something that was more sort of clinical and entomological, like be sure to check for ticks and they're the size of a poppy seed. And so asking what kinds of prevention messaging would really be effective and get good uptake by people. So this is human behavioral science, and we have a variety of colleagues in life sciences communications who are helping us tremendously with this work. We had people who were enrolled on the East Coast and people who were enrolled in the Midwest, and what we found was that Wisconsin users of the Tick app tend to be more outdoorsy and be doing things like hunting and hiking and camping. It turns out that many more people in the Midwest mow their own lawn, which we wouldn't have expected. And what we're also seeing is that because of the work we're doing with surveillance, the that prevailing sentiment that you're going to come into contact when you're out recreating in natural areas is less true. And so people are finding ticks more in and around their own homes. We can see that data in the tick app, but we can also see that with the surveillance that we're doing in Wisconsin and in states around us. So really important information. We also can see that Wisconsinites tend to check for ticks more than they do in the Northeast. So Wisconsinites are shown here in gray. They're more likely to wear repellent. They may be more likely to like tuck their socks in so adjust their clothing, and perhaps to shower after being outdoors. And so again, this is really nice because it's actually congruent with our knowledge, attitudes, and practice study that we were doing with our colleagues at the Minnesota Department of Health. All right, so in 2018, so now I've got the years right. In 2018, we had participants that were largely in and around Wisconsin and then around New York. It's in particular Maria Duke-Wasser's lab at Columbia that we're working with on this project. And in 2019, we saw broader geographical enrollment. And so, you know, people are scrolling through the App Store and they find something about a tick and they put it on their phone. So we doubled our users in 2019. This is also a function, I hope, of the great recruitment work that our postdocs were doing to make this happen. And so as the tick app is evolving, you know, we designed it to be a behavioral assessment tool. We're seeing that it's evolving into something more, perhaps a venue where we can provide educational material. We also let people know when tick activity is likely to be high. So we have phenological data from our different partner agencies, and you could look at a sort of scale that shows you on a green, yellow, red scale what the tick risk at that time of year is in your area. We provide tick identification, so people can send us actually a picture of their tick, and we have professional tick identifiers associated with our project. So people whose job it is to look at those photos and respond to those tick photos. We provide tick check reminders. So this is the public health intervention part, and then that intervention messaging. So the tick app has been a point of pride for our center, and I think we are learning a lot about human behavior in contact with ticks. We have a couple of publications on this already, again, from our great postdocs, Bina Cabron and Pilar Fernandez in particular. And this work is work that we do in total collaboration with Maria Duke-Wasser and Jean Tsao, with, of course, Susan as the UW-Madison tick expert. We also, of course, do work on mosquitoes and mosquito-borne disease. And so we likewise have a variety of projects associated with different areas of emphasis: surveillance, outbreak responses, taxonomy, data science, etc. So I'm just going to go through a few of those. When the center started and everyone was really concerned about Zika virus, of course, we needed to know if, in fact, Aedes albopictus was in our region. And so dug in and tried to learn more about Aedes albopictus invasion biology. We know that it's a mosquito that's really adept at invasion. And so it's indigenous to Southeast Asia and China, and by the 1950s, it had moved around the world, probably with movement of like ship ballast and goods where there would be water contained. It's then moved into the U.S. and Central and South America in the 80s and 90s, and so it's really made its way all over the world. So this is called the Asian tiger mosquito. You may have heard of it. It's a horrible pest. So not only is it in the lab, at least, shown to transmit at least 20 plus different kinds of pathogens, it's also one that's incredible in its nuisance potential. So it's a daytime feeder, and they are voracious feeders. I have them in my lab, and I get anywhere near the cage and they're all up against the screen trying to feed on me. So really, really voracious feeders. And they're so invasive because they can move around with their desiccation resistant eggs in small containers. People find them in things like happy bamboo that's moved around. And we see that there's been globally repeat introductions with the movement of military equipment and commercial products, and in particular tires. Tires are a really important source of these mosquitoes. So all over the 5-state region, we started looking more for Aedes albopictus. It was already fairly well established in southern Illinois when the center started, but elsewhere there had been reports of yearly invasions in a couple of places, but really not establishment of the mosquito. So we had an approach where we used BG sentinel traps, which have been designed for, in particular, for what we call anthropophilic mosquitoes that are particularly attracted to people, and also these oviposition cups. So this is super simple, where we have just a black cup with a tongue depressor in it, and mosquitoes that are what we call container breeders will come and lay their eggs on the tongue depressor. Then you can pull the tongue depressor and look to see what mosquitoes have laid their eggs there. In 2016, we did surveillance with public health partners. Then in 2017, we decided to get more official with going to particular places. So with public health partners, they were setting these oviposition traps in their backyards. With our more formalized approach, we decided to go to a variety of counties and particularly target tire yards. So tire recycling facilities or tire sales, right? So places that sell large numbers of tires and would have tires stored outdoors. We could see this actually using Google Earth. And so we used an approach that was both searching for tire purveyors, like in the white pages, and then also looking via Google Earth for evidence of, a mass of little black circles on the ground. So we found places like this facility. You can see they actually have massive tires, these agricultural tires that are used for tractors, etc. So they have these big stacks of those tires, and you can see that they're right up against a suburban neighborhood. And in 2017, at that site that I've just shown you, and also actually in and around Madison, we did in fact find tongue depressors, so those little sticks that we put into the ovitraps with Aedes albopictus eggs on them. So we found them in Dane County, where Madison is, and we found them in Waukesha County, which is over here by Milwaukee. Not very many, but enough that there's concern. So we could say, oh dear, there are Zika vectors potentially invading Wisconsin. Right. So we actually continue to find at that site those mosquitoes, which is really distressing. We were hoping this was just, a happenstance sort of not invasion and establishment, but just movement of tires into Wisconsin. And so it was maybe a one-time event, but we go back every year and we keep finding that mosquito every year. And so we want to know, where are they coming from using haplotype assessment. In collaboration with some colleagues, we want to know if the eggs will survive over Wisconsin winters. So we're doing projects where we set out tires and monitors for temperature and humidity to see whether the eggs survive in our cold winters. And we want to know how we could control them in a really efficient way. So we've been using larvicides inside tires to assess control potential. We've been using spinosad, and seems to work really, really well. And we have to figure out ways to transfer that technology to those businesses. So it's quite easy to do. We could train them, and they could, I think, affect really good mosquito control on their own without having to hire anybody or to beg us to come over and help them. So hopefully a win-win. We also have been working on outbreak responses. So part of the idea behind the centers was that we should have these really nimble entities where if there's an outbreak, we could rapidly respond. And I think we've done this pretty well. We have some points of pride there. For example, when the hurricanes hit in, I guess, 2017, in and around the Houston area, we got calls from people who were working in mosquito control, and they said, we are trying to understand how many mosquitoes are here and whether or not our controls are working. And we do not have personnel enough to help us get this done. And so within just 3 days, we took some of the people that we'd been training, got them briefly employed with that agency, got them plane tickets and sent them to Houston. And they spent a week there and started cranking through mosquito samples. And they took a job that was taking all day down to being done before noon. And so it just felt really satisfying that the people we were working with on surveillance and mosquito identification in Wisconsin were confident enough to just mobilize and go and do that important work to the benefit of public health. Another example of an outbreak response is what we've done with Jamestown Canyon virus. So this is a Bunya virus. If you're into viruses, there are Bunya viruses that are transmitted by thrips. So maybe some of you know about like tomato spotted wilt virus. Jamestown Canyon virus is related, and it's a virus that is neuroinvasive. And so people who get Jamestown Canyon virus and show signs of disease get really, really ill. And it can lead to either death or what we call long-term sequelae, where people suffer from cognitive issues as a result of having inflammation on the brain. So we really want to understand what mosquitoes are involved in the transmission of this virus and what we might do to intervene and control and protect people from getting it. So we decided to do some intensive mosquito surveillance, going out and looking for what mosquitoes are there and whether they're infected with the virus, and then also doing laboratory vector competence studies, because frankly, we don't know well what mosquitoes are involved in transmitting this virus. If you look at the literature, there's a list of like 20 species that have been associated with Jamestown Canyon, but that doesn't always mean then that you're looking that list is really useful in a particular place and space, depending on the mosquito communities there. So there was this profound increase in the number of cases of Jamestown Canyon virus in 2017, far exceeding what we get for West Nile cases. And the state Department of Public Health asked us to please intervene and do something. So we went out and looked for mosquitoes. We went to a particular hotspot in Sawyer County in northern Wisconsin, and we hit multiple sites. We went to case sites to see where people were when they got infected. And we saw things like these bogs. We saw ephemeral pools. We saw places where people were living in homes that were just surrounded by all kinds of mosquito harborage, right? Like if you look at this, you can see all kinds of places where water might sit and mosquitoes might breed. And these tires were just loaded with mosquito larvae. So understanding what puts people at risk for contracting Jamestown Canyon virus is really important. And this particular county where we went to work, we were working in and around Hayward, Wisconsin, which is like the muskie capital of the world. In case that's your jam and you'd like that, you could visit Hayward. And it's immediately adjacent to a reservation, the Coeur d'Alene reservation. And so we were working with local public health and Indian Health Services. And this is a part of Wisconsin that is socioeconomically really challenged. So it's ranked very low for health outcomes and social and economic factors. And those things tend to go hand in hand with some of the mosquito-borne illnesses. We threw everything at it to try and understand what mosquitoes were there. We used what's called a Shannon trap, which we actually just used blacklight and did some nighttime trapping. Sometimes you could put like a tasty host inside this little tent and trap mosquitoes that way too. We didn't do that. We can use CO2 baited traps. This is called a resting box. So you trap mosquitoes that are full of blood and need a place to rest to digest their meal. We use these backpack aspirators and BG Sentinel traps, and also just our own eyes to find mosquitoes. We also used emergence traps. So we borrowed these from a colleague in entomology who does emergence for potatoes, and we floated them on ephemeral ponds and collected all kinds of mosquitoes that way. And we brought the mosquitoes back and identified them to species, which is no small feat, and then figured out which ones were infected with the virus. And what we kept finding year after year was this particular mosquito, Aedes provocans, that was infected. So we found repeated years that the mosquito was infected with the virus. And in fact, we even found some newly emerged mosquitoes that were infected, which suggests that mosquito might be involved in transovarial transmission of the virus. Then we worked with colleagues at the CDC to get full genome sequence of that virus. virus, isolated the virus so we can continue to work with it. And what we found was that the mosquitoes that are present have very distinct phenologies. And so the one we find infected with Jamestown Canyon is really an early season mosquito, and a mosquito that clearly wanes. And so there's got to be other mosquitoes that are getting the virus from infected hosts and then transmitting it for the duration of the season. So this is a fun epidemiological puzzle. We also trapped all kinds of mosquitoes and brought them back to the lab so that we can take them and infect them with virus. We did smart things like these are obviously larval collections. We bring them back and let them emerge in the lab. We also tried to get adults, and so we would trap them in our CDC traps, and then we found that they were all dying on the transit back to Madison, so we started trapping them directly into these large mosquito cages so we could bring them back and infect them, and those results are pending. So we're doing those exposures. Actually, we're doing some of those exposures right now in the lab. We're also working on some projects to understand whether mosquito control works or not. So if we're working with public health partners and they are trying to control mosquitoes, so is it worth the investment? That's the question. So it's a massive endeavor to control mosquitoes as larvae and adults. It's something that very few public health agencies have capacity to do. And the question is, what's the bang for the buck there? So what we've set out to do with some of our graduate students is better understand whether larval control impacts West Nile transmission. We've been doing this work in Milwaukee and Chicago. Most of the mosquito control that goes on across the US involves some kind of what we call catch basin treatments. So catch basins are these water runoff catchment traps under the ground. You usually see some kind of grate over them. They collect water and try to settle out leaves and other things so that kind of organic effluent doesn't go into streams and lakes. In so doing, they create this perfect sort of really rich organic place where mosquitoes can breed. And in particular, the mosquitoes that transmit West Nile really like that muck underneath the water, underneath the streets. And so trapping and controlling mosquitoes that transmit West Nile often involves controlling them in these catch basins. So the question is, does it work? And so we worked in an area in Milwaukee to do this. Our null hypothesis was that catch basin larvicide treatment has no impact. And so this is the question. Is it worth investing in a whole bunch of water-soluble packets of BT to throw down into these catch basins to control Culex pipiens? And so our graduate student Haley Johnson created a really lovely sort of stepped wedge design to treat and leave untreated certain sites across the Milwaukee area where there had never been any of this kind of mosquito treatment before. And just briefly, what we're showing or what we're seeing is she did the treatments and then she did really intensive trapping for adult mosquitoes. And the data are, of course, messy because this is biology. But what we see in treated and in untreated places is really similar numbers of adult mosquitoes, which suggests that catch basin treatments may not be getting at all of the point sources for mosquito breeding. So if you're going to do catch basin treatment, you may not see an effect. So those are the kinds of things we need to understand better. We will also want to understand if adult control, so this is ultra low volume spraying, you may have seen this in your neighborhoods. We want to understand if that likewise has impacts on adult mosquitoes and therefore transmission on West Nile. This is work that's being done by Christina Lopez in our center. And so Christina and colleagues at the Northwest Mosquito Abatement District have been doing this widespread mosquito treatment and asking whether it impacts the numbers of mosquitoes that are out there. She's done this work in a way where she's trapping mosquitoes, so she gets abundance information, and she's checking parity status. So for those mosquitoes that are intact, she pulls ovaries and looks for evidence of them having had a blood meal and laying a batch of eggs. That tells us something about whether they're likely infected with West Nile and if they could transmit. because the only way they get West Nile is by taking blood. So we want to understand the parity as a function of age structure and risk associated with transmitting West Nile. So she ran all kinds of traps for every night during the week. This is really super intensive work, and asked then whether those mosquitoes had any particular age structure and whether they were infected with West Nile. And so I'll just show you a snapshot of the data to show that on the left, you see sites where control mosquito, or there was mosquito control, or sorry, this is the control. This is the control sites where there was no mosquito control. So we should use different terminology there. And then we had the treated sites. And what you're seeing is the proportion of nulliparous mosquitoes, so mosquitoes that had not taken a blood meal. And you can see sort of a shift in the dynamic where in places where there was no mosquito control, there were fewer nulliparous mosquitoes, so more mosquitoes that had blood fed, and the opposite in the treatment areas. So this is really important. If in the treated areas you're shifting the age structure of the mosquitoes and you're having an effect in such a way that your mosquitoes are not blood fed, then you should see some difference in West Nile infection rates, and she is starting to see that. We're also finding all these offshoots projects, and so we've done a variety of different and important things, I think, with mosquito taxonomy. I know that often entomologists think that mosquitoes are exceptionally well studied, and that's true in some sense, but I think the taxonomy is still lagging. And well, I know that's true for sure. And there's lots of need for revision and better keys and better identification tools. One of the things that we've been up against for a long time is that mosquitoes in the Culex pipiens, Culex restuans space are really difficult to tell apart. And it's probably pipiens that's more important for transmitting the virus to people. And so it would be nice to be able to differentiate the adults. And so I had a student working with me who is a bona fide mosquito systematist, and she came up with some really useful characters for separating out those species of mosquito. We also have now a key to the black-legged mosquitoes that transmit Jamestown Canyon virus. So I think these are really nice, again, sort of added benefits of the work we've done as a center. We also have discovered a new species of mosquito, so hopefully we'll be able to put that publication out soon. We also have, as an effort across the center, we're trying to get all of those records of mosquito and tick encounters into a particular repository so that then we can do deeper dives into the ecology and biology of those species. So our vector records repository is coming together as well. And so all of this is made possible because we work with trainees. I showed you how we are doing applied work, how we're working in communities that practice with public health by doing sort of outbreak response work, for example. And all of it's possible because we're working with a whole cadre of really enthusiastic and wonderful trainees. And so I'm going to end today just by talking a little bit about our training process. We do train public health entomologists, and our concept for training was that we wanted to train people at the undergraduate, graduate, postdoc, MPH, so Masters of Public Health, all kinds of professional students and existing practitioners in public health entomology. We wanted to provide short-term sort of workshop kind of trainings, but also immersive experiences. And so we set up our budget for the Center of Excellence so that we could support fellowships. And now what we do is we support something like 20 people to work across the center with our different partners and do all kinds of really invaluable kinds of contributions to our research and surveillance efforts. So it's such a win-win because the students get to have a full and authentic experience in working in vector control or surveillance, or even in labs doing microbiome analysis. They get really intense and authentic vector biology experiences. And then we have the benefit of having our field teams populated by these enthusiastic and great people. We also have a concept where we have created something called a public health entomology certificate. I know ESA is also doing this now too. Our entomology, our PhD certificate comes from our Center of Excellence. So rather than having to do something sort of transcriptable at the university, we created a certificate where anybody in the center in our 5-state region can get it. So we came up with sort of areas of competence, and everybody has to take classes in medical entomology and have pesticide applicator licensure, and they can do that in a 5-state region. So they don't have to take classes at UW-Madison in order to get our public health entomology certificate. And I think this has been extremely successful. So I'll just kind of cruise through our fellowship training. This last summer was interesting because we had to adapt to COVID-19. And so we had people working in all kinds of different places and at home. So we sent them a kit so they could learn how to be public health entomologists. So you can see these kits. We worked with them for 8 weeks to train them up in public health entomology. And then actually we were allowed to deploy some of them to our field crews and into our labs so that they could get this experience despite the pandemic. And I'm happy to talk more about that as well. So with that, I have more slides about our training, and perhaps the grad students would like to hear more about that, or somebody else. I know Purdue has this legacy of excellence in outreach and training. And so if that's something that's exciting to you, I'd love to tell you more about how we adapted to online experiences for summertime work for our students. And so the Midwest Center of Excellence really is this massive partnership, and it's fueled by a lot of outstanding trainees, and just not possible without our partners in public health and vector control. So I'll stop there and invite your questions. And as I said, I couldn't go too deep on any of those, but I'm really happy to talk more about them if you're interested in a particular project. All right.
Thank you, Lyric. Thank you very much for a really informative and excellent overview of the activities of the center and the really exciting research that you and your partners are conducting. I would encourage our attendees to please submit any questions for Lyric through the Q&A mechanism. I'm monitoring that, and we'll be happy to pass your questions to Lyric. I actually have a question myself. I thought I'd kick us off with.
Yeah, sure.
You're monitoring so much. You're monitoring diseases, pathogens, parasites, and you're monitoring the vectors. And there's an awful lot to keep track of. You're looking at what's going on with the black-legged tick. You're looking at invasion by Aedes albopictus. So I just wanted to ask about one more. Are you also looking for the Asian longhorn tick? Is that something that's on your radar? And it's something that's on ours, as is the Gulf Coast tick.
Yeah.
We're watching those two ticks very, very carefully.
Yes.
I don't want to give you anything more to monitor, but I just was curious about that.
Yeah, so certainly in a state like Wisconsin, that is the dairy state, where we have lots and lots of cattle, we're very concerned about the Asian longhorn tick. So for those of you who don't know about this tick, it's a tick that's native range is really in Southeast Asia, and it's made its way over here, and it's parthenogenic, and it can replicate in these massive numbers. And I've seen videos from colleagues over on the East Coast, like in Pennsylvania, where the ticks are just swarming. It's just this disgusting biomass of writhing ticks on cars even, and so many on little lambs that they actually get exsanguinated. So it's a really horrible tick, and we're definitely concerned about how it'll move across the United States. And I think states are progressively westward seeing that tick. And so we certainly have it on our radar, Kate, but I think it's probably going to be slower to come this far north. That said, we have Illinois as part of our center, and so They're on the alert. Illinois Natural History Survey is part of our center, and so Holly Tootin there is watching out for that tick, as well as the Gulf Coast tick, and she's been doing a concerted surveillance effort in Illinois to find Gulf Coast ticks as well. So sort of to keep people alerted to the to the longhorn tick. We've been working in community with veterinarians. So luckily I'm part of a vet school. And some of the agencies that track veterinary pathogens and ectoparasites, and have offered some training so that people know what the tick looks like. And I think people are starting to see that we're a resource that they could come to. I mean, already they knew about all of us as land grant entities, but I think the center has gained more traction and credibility, and so they do seek us out with questions about the longhorn tick as well.
Yeah, thank you. I know well, it's been found in Kentucky, so it's moving, and
it's moving.
And then the Gulf Coast tick, Indiana State Department of Health is reporting that. So
yes, yeah, I heard from them just last week on a call. So one of the things we do as a center is, in order to kind of maintain our community, we have a weekly all hands call, and states around our area, including Indiana and Ohio, tell us about the kinds of things they're up against. So it's really nice because we're starting to hear, we're just doing a better job of communicating. So, we would hear like, okay, Indiana's starting to see this, and that informs practice in the surrounding states.
Thank you. I'd like to give you a question from Elizabeth Barnes. She's asking, have you encountered any, this is a good question. I'd be very interested to see your response to this. Have you encountered any resistance to tick or mosquito control efforts during your outreach programming? For example, resistance to pesticide applications, and if so, how have you handled it?
Oh, good. So this isn't actually about the physiological response. We are actually monitoring for resistance to the pesticides, and see shockingly more of it than we had expected. But you're talking about public perceptions, and one of our colleagues in the Northwest Mosquito Abatement District has done a really nice job of trying to understand public perception of mosquito control. There's certainly lots of concern about this related to pollinators and biodiversity. And what he has is actually, and a lot of mosquito abatement districts have this, is a no spray zone option. So there are people who will say, I do not want your spray to hit my property. And they can elect to not be in a spray zone. And then the trucks have to shut off the sprayers when they're going around that house. And oftentimes this is Dr. Patrick Irwin, who runs that district. What he's found is that there is opposition, but then oftentimes when he has outreach events and he'll talk to people about the spray, and he'll talk about how it's sprayed in the evenings and it's ultra low volume, and there are lots of things about the logistics of spray and the physics of spray that make it so it's really unlikely to hit a pollinator, then he'll find that people are more amenable to it. So hopefully that starts to get at your question. And I think there's certainly merit to better understanding if that resistance is associated with just concern about chemicals, or if it's concern about, as again, pollinators, or where that fear and concern comes from.
I know you're speaking this afternoon with Dr. Brock Harper, who will probably share some very interesting experiences in the state of Indiana with the response to eastern equine and the attempts to control that in the upper Midwest, Indiana and Michigan in particular. Very different situation, obviously aerial spraying, and it's tricky, very tricky.
It is very tricky, and it's such a dangerous. So you really have to weigh public health, and sometimes public health, outweighs to the distress of people who are concerned about biodiversity and environmental wellness.
And how you inform people of your
absolutely
for spray applications. And give them the opportunity to respond. So another question here from Carlos Brijosiero, who is a graduate student. This is in reference to Aedes albopictus. And he's asking, is albopictus the preferred vector of Jamestown Canyon? I guess it actually probably means primary, assuming the primary vector. And it does, do you know if it somehow impacts the mosquito? And have you looked for viral titer in the salivary glands with the mosquitoes?
It was super fun. This is all the kind of work that we are hoping to do. I don't think that Aedes albopictus is a primary or preferred vector for Jamestown Canyon virus, but certainly important in terms of things like chikungunya and dengue and other viruses. We actually don't test albopictus in this area for infection with arboviruses. The risk is quite low, so our numbers of the mosquito are quite low, and we know that our imported cases of travel acquired cases of chikungunya and dengue virus, for example, are really low. So the likelihood that the mosquito at this population level would transmit those viruses doesn't encourage us to invest in the testing at this stage. The work on understanding how those viruses impact the mosquito is really interesting to me, and we've just started to take some of our field caught mosquitoes, not Aedes albopictus, but things like Aedes japonicus and Aedes provocans and Aedes canadensis, so some of those other field collected mosquitoes, to see how competent they are in the lab. And indeed, when we do those kinds of studies, we'll be asking things like, what is the propensity of the virus to move to the salivary glands? And we haven't actually monitored the titers of virus in the salivary gland directly, but we get that by proxy by having the mosquitoes spit into a capillary tube. Then we can take the saliva actually from the capillary tube and put that into cell culture and monitor the titers that way. So that's work we will be doing.
Okay, great, thank you. Another question here is, if you could comment, Lyric, how the regional centers work together. I mean, how do you identify your priorities and how do you collaborate, I guess, on those priorities? Because there's some of the things that you're responding to in the Midwest Center are similar to what might be experienced, say, for the Northeast, but very, very different to the South.
It's something the CDC would really like to have us do, is make sure that we're working in community with our colleagues in other centers of excellence. As a way to practice that, we try and get people into our weekly calls from across the different centers so we can come up with collaborative ideas. For example, the PacVec Center at UC Davis has done a really interesting project where they're looking at modeling where Aedes aegypti and Aedes albopictus might be. So they have this Aedes modeling project, and they came and talked to everybody about getting data on those invasive mosquito species, and we've all shared our data, and so they've been incorporating that into their models so that we can better understand what the range of those mosquitoes might be. So that's one collaborative endeavor and kind of an example of how we got in on it. We just recently had a call with our centers, all of our center directors. So we get together with some frequency, a couple times a year, to talk about what people are doing and what concerns people are responding to. We also have the TIC app as another example of a cross-center kind of collaboration where we really wanted to work with this entity on campus on a TIC app. And then we, in collaboration with Maria Duke-Wasser and work she had done already on a TIC app, came up with a joint project. And so, that comes just kind of naturally by our connections to our colleagues in those different centers. It also comes as a function of need and needs assessment that are done by the different centers. So I think, the great thing about our vector biology community is that we do tend to be quite collegial and receptive to collaborative work. And that's so facilitated by having resources through the center to do it. It's so nice to have funding to pay for, postdocs who can go and make a TICAP happen. So having the support from the CDC for these centers has been just outstanding.
I hear you on that. After working for many years with, county and state agencies, and seeing how they struggle
Yep.
to be able to cover not only mosquito-borne diseases but also tick-borne diseases. It just highlights the important role that you're playing. And it's also wonderful to see the coordination across so many agencies and entities in your center and others. So kudos to you guys. And
yeah, and to the CDC for having the vision, right? Like, I think it was a really visionary idea, and I think it's manifest in a really powerful way.
Great. And I have many questions for you about the CDC, which has been has taken some heat recently given its response to an infectious disease outbreak, not vector-borne, thankfully. And I sort of unfortunately, I think the response has been challenged. And yet, CDC plays such an important role in the response to vector-borne disesos. And so a very interesting thing that perhaps we can follow up on. I know we've come up on our time. So on behalf of everybody, I would like to thank you so much for your presentation. I know you have some slides that you might share with the graduate students over lunch. I'm sure they'd be very interested to continue that conversation. And for others who are here and perhaps haven't, if you have a question that comes to mind after the presentation, you're welcome to email it to me or to, I'm sure that Dr. Bartholomay would be very happy to respond as well.
Absolutely.
So thank you so much. And I'll clap.
Thank you. Thank you.
All right. Thanks so much, Larry.
Yep. All right. Thank you, everybody.